everyone, welcome to Drishti CUET and welcome to your biology revision classes. So I hope guys you all are doing great and you have completed your syllabus obviously, and I hope till the time you have revised your syllabus also 2 or 3 times. See, before going to the exam, just try to revise your syllabus at least 3 times, right? Minimum 3 times, so you have to revise the syllabus, right? Now here I am also going to help you in your revision section, and how I am going to help you, let me explain. Here I am going to revise your whole syllabus in the form of your solving sample papers, right? And we are also revising your whole syllabus based on your previous year question paper, and also will solve your MCQ type CUET previous year paper. So based on that, we are going to solve all the type of questions. We are going to practice, we are going to revise all the type of questions here. Okay, so this is the very first session, and here first of all we are going to revise the syllabus based on your CBSE sample paper. All right, so first of all we'll practice or we'll revise the syllabus through sample paper, then through previous year questions, and then through your CUET previous year questions. All right, now based on the sample paper, here I am going to tell you also like how you have to write the answers in your board exams as well, right? Like if you have any question, so how much you have to write, what are the pointers you must include in those points, right? So all these things I am going to explain you in this particular session. All right, so are you all ready? Shall we start the session? So first of all, let us look over the pattern of your board exam paper, right? So these are the patterns you may also get whenever you will get the paper now. This is how your paper first page looks like. So don't get panic कि कैसा होगा paper. This is the first page of your paper, okay? And your paper consists of 70 marks. Like if you are talking about physics, chemistry, biology, all the theoretical paper consists of 70 marks. The remaining 30 marks includes your practicals, okay? So out of 70, your marks judged, and the time is given 3 hours. So just try to utilize the 3 hours, whole 3 hours. Sometimes what happens, the students writing very fast, skipping the points, and they just try to finish the paper early, like within 2 hours they just try to finish up the paper, and then remaining 1 hour they are just still, because teacher do not allow you to move out of the class till 3 hours completed. Okay, so just utilize your time, give time to each and every questions, right? And analyze the question and then try to write the answers. All right, so here the general instructions given on your paper, that all the questions are compulsory. That means total 33 questions you have in your paper, and all 33 questions are compulsory. It's not like out of 33 you have to solve 23 question, or you have to solve only you have to solve 30 questions like that. No. Yes, within the questions definitely you will get the options, right? Within the questions you will get the option, but not like out of 33 you have to solve all the 33 questions. After that, the questions paper has 5 sections and 33 questions. So total in your board exam you have a 5 section, and out of 5 section 33 question is given. Section A is very important and very scoring nowadays. Why? Because it consists of 16 question, 1 mark each. 16 question means here you will get the MCQ based question as well as your assertion and reasoning type question. So if you score 16 out of 16 na, it will boost your marks, राइट और यहीं पे अगर गलती हो जाती है ना, तो आपका मार्क्स जो है ना ग्राफ एकदम से गिर जाता है. So you have to take care of this 16 questions. Okay, these are MCQ based and your assertion and reasoning based question. Then section B has 5 questions, and these 5 questions are of 2 marks each. So now the second section, section B, is of 5 questions, and each question is of 2 marks. That means 10 marks of section B. Section C has 7 questions and 3 marks for each. So approximately 21 marks questions you will get in the section C. And then section D has 2 questions, and these 2 questions are case-based question. So nowadays case-based questions are very, the CBSE has introduced the case-based questions just to prepare you for your further competitive exams, right? Like when you will opt CO2 CUET exam also, na, so there also you will get this case-based question. Okay, so you have to be prepared with this case-based question, and like 4 marks. Each question 4 marks, so total 8 marks you have section D. Section E has 3 questions and these are 5 marks. That means 15 marks of your section E. Okay, so these are the division of the section. No choice, all 33 questions you have to solve. Internal choices has been given. Student has to attempt only the alternative questions, right? And wherever it is required, try to make a neat and clean diagram as well. So this is just the pattern, right, of your question paper board exam 2026. All right, so let's begin with the questions, guys. I hope till now you have revised all your syllabus as well. Okay. Okay, so let us go with the first section, that is section A. Number of section is like 1 to 12 are multiple. So in section A you have a 16 question. Out of 16, 12 question is MCQ based and the remaining is assertion and reasoning based question. So here 1 to 12 is your MCQ. Only one is the correct choice. Select and write the correct choice as well as answer to this question. Now abhi board exam mein, how you have to write down the answer? You have to, like, suppose this is the first question, you read the question, you know the correct answer, then in your answer sheet, okay, answer sheet mein how you have to answer? In answer sheet, question number 1, like answer number 1, whatever will be the option, like suppose for this question the C option is the correct one, so write down the option plus whatever is given in the option, like mitotic division of nucleus of generative cell, right? I'm giving you just example how you have to solve here. Okay, so what you can do, this is how you have to write down the answers for your MCQ questions. All right, clear? So now abhi start karte hain revision. Okay. So this is the first question. The male gametes in the plants are formed by. The male gametes in plants are formed by mitotic division of nucleus of vegetative cell, meiotic division of nucleus of vegetative cell, mitotic division of nucleus of generative cells, and the meiotic division of nucleus of generative cells. So what is the correct answer here? See, फ्लो में मैंने करेक्ट आंसर आपको बता ही दिया है, बट लेट मी एक्सप्लेन यू दिस क्वेश्चन फर्स्ट ऑफ ऑल. See, in case of plants, right, in case of plants, male what are male gametophytes, guys? Here the question is male gametes. So now we have male gametes in the plants are formed by mitotic division of nucleus of generative cells. So you know that in case of plants, the male gametophytes are what? Your pollen grain. Okay, and if you will see the structure of pollen grain, how it looks like, it has a outer covering, right? It has a outer covering which is made up of a chemical compound known as sporolanin. So this is what? Sporolanin. Remember, this sporolanin is a enzyme resistant, temperature resistant, pH resistant. That means नथिंग विल इफेक्ट हियर, ठीक है, किसी चीज का भी इफेक्ट नहीं होता है, और दैट्स व्हाई वी कैन यूज pollen grain as a fossils. Okay, now this is the pollen grain, right? This pollen grain has two cells within them, right? One, it has a generative cells, and one, it has a vegetative cells. So this is what, this is the vegetative cell. This is the vegetative cells of pollen grain, and this is your generative cells within the pollen grain. ठीक है, ये हमारा generative cells होते हैं. Now the vegetative cell is used to provide nutrition. Basically यहाँ पे food store होता है. Whatever the food is storage, that all in the pollen grain vegetative cell. Generative cells further undergo mitotic division. ये what is the difference between mitotic and meiotic division, guys? See, meiotic division occurs in the germ cells. ठीक है, meiosis is meiotic. Germ cells, within the germ cells you will get the meiotic division, and the cell divides into 4. Ultimately you will get the 4 daughter cell, but generative cells divide into 2 here with the help of mitotic division. So when the generative cells undergo mitotic division, you will get 2 male nuclei, right? So this generative cell divided mitotically, and you will get the 2 male nuclei or 2 sperm nuclei. So meiosis कहाँ हो रहा है? Where does this meiosis generation or you can say where does this meiosis takes place within the plant? Meiosis कहाँ हो रहा है? Meiosis occurs earlier during the microsporogenesis. So meiotic division takes place during microsporogenesis. ठीक है, तो माइक्रोस्पोरोजेनेसिस के टाइम पे meiotic division हुआ, and this microsporogenesis, with the help of meiosis, you will get 4 microspore, and these microspore ultimately converted into pollen grain. Now within this pollen grain you have 2 cells, vegetative, generative. Then this generative cells further undergo mitotic division, and you will get 2 male nuclei. Clear? Any doubt, anybody? Yes, quickly. So here the correct answer is your C, meiotic division of nucleus of generative cells. So the male gametes in the plant are formed by mitotic division of nucleus. Now the second question. So how you have to write down the answer in your board exam copy? 1, option C, and then you have to write down the whole option. Okay, then suppose let's go with the second question. The primary endosperm nucleus is formed by fusion of which of the following? Primary endosperm. When you will get this primary endosperm? A male gamete and a female gamete, a male gamete and 2 polar nuclei, a female gamete and 2 synergid, 2 male gamete and a egg cell. So remember, this question is based on your double fertilization. Okay? Do you know the answer? What will be the correct answer here? See, this is based on the double fertilization. Let me tell you the concept of double fertilization, guys. So, in case of double fertilization, what happened during double fertilization? See, suppose this is the female reproductive structure called pistil, right? Within the pistil, this outer structure is called stigma, this is called style, and this is called ovary. Okay, within the ovary you have a ovule. Now what are these structures? These are called ovule. Within the ovule you have a embryo sac. Within the embryo sac you have a egg. Okay, so now what happened here? Within the ovule only you have a embryo sac, है ना. और वो कैसा होता है embryo sac हमारा? Here you have 3 antipodal cells, then you have 2 polar nuclei, and you have 1 egg, right, and 2 synergid cell. This is how the structure of embryo. Okay, then what happens? Suppose the pollen grain somehow reaches. Now what happened? The pollen grain, this is what? This is the pollen grain. With the help of pollinating agent, the pollen grain stick to the surface of stigma. Now from the pollen grain, a tube generated called pollen tube, and this pollen tube enters into the ovule. So what is this? This is called pollen tube. From the pollen grains, pollen tube generated through the germ pore. Remember, we have an area over the pollen grain known as germ pore. Okay. Now what happened? Within the germ pore, right, here we have 2 male nuclei. Now what is this? These are the 2 male nuclei, right? What are these? These are the 2 male nuclei. How does this 2 male nuclei comes? So देखो यहीं पे ये जब divide होंगे, here you will get 2 male nuclei. Now here you have an area, okay, where you have no sporal anon, and this particular area is called germ pore. Through this germ pore only the pollen tube arises. Okay, and then the 2 male nuclei enters through into the pollen tube. Okay, 2 male nuclei you will get. Now what happened here in the fusion? So now the first male nuclei, right, through the pollen tube enters into the ovule. ठीक है, तो अभी क्या होगा? First male nuclei enters into the ovule and the male nuclei meets with the egg nuclei. ठीक है, here you have a egg nuclei. So the male nuclei fuses with egg nuclei, right? And this forms what? This process is called syngamy, and ultimately what you will get? You will get zygote. Okay, so now here the male gamete, the male nuclei is also haploid, having n number. Egg nuclei is also what? Haploid, having n number, right? And ultimately you will get zygote, which is diploid. So this is the first male nuclei. Now what about the second male nuclei? What about the second male nuclei, guys? Second male nuclei, okay, which is also haploid. ये जो second male nuclei है, ये भी तो haploid ही है. All right. So now this second male nuclei fuses with these 2 polar nuclei. What are these? These are the 2 polar nuclei. So how many polar nuclei we have? 2 polar nuclei. So the second male nuclei fuses with 2 polar nuclei, right? So haploid. Now 2 nuclei, so it is called as a diploid. And here 3 nucleus now, 1 male nucleus and 2 polar nucleus fuses together. So this fusion is called as a triple fusion, and ultimately as a result you will get endosperm. So what you will get as a result? Endosperm. All right. So now this endosperm is what? Having 3N triploid. Now this endosperm will provide nutrition to zygote, and this zygote converted into embryo, and then embryo converted into seed. So like this the process is going on. Okay, so now what is the question here? The primary endosperm is formed by the fusion of which following? So male gamete and a female gamete? No. When male and female gamete fuses, you will get what? Zygote. Male gamete and two polar nuclei. So this will be your correct option. Alright. Now let us move to the third question. During the menstrual cycle of human female, now menstrual cycle, formation of Graafian follicle is stimulated by the secretion of which of the gonadotropin hormone? Now guys, remember, you know that the menstrual cycle में 4 phases होते हैं, right? You have a bleeding phase, first menstrual phase, then you have a follicular phase, luteal phase, right? These are the phases you have. Ovulatory phase, right? So these are the 4 phases you have. Now in the follicular phase, there will be a formation of follicles. You know, first of all you have a primary follicle, then secondary follicle, tertiary follicle, characteristic having antrum, a fluid filled cavity, and then tertiary follicle converted into a Graafian follicle. Now this Graafian follicle bursts and the egg reaches out, ova reaches out, enters into the fallopian tube. So the Graafian follicles, whenever the Graafian follicle forms, at that time the level of FSH and LH increases. Okay, so these hormones stimulate the formation of Graafian follicle. तो अभी human reproduction में this topic is very important. आपको ये hormones याद रखने हैं कि at which phase which hormone releases. Like during ovulation, जब egg बाहर निकलता है तो LH का concentration बहुत बढ़ जाता है. So like this you have to prepare. Then the next is the experimental proof on the thermal stability of genetic material was first provided by. Now this is your topic related to the molecular basis of inheritance, and here you have learned about the Griffith transformation experiment. Remember, Griffith का आपने transformation experiment पढ़ा. You have learned about the Hershey and Chase experiment. Okay. Also you have learned about the Michelson and Stoll experiment, right? And Jacob and Monod को भी आपने पढ़ा था. So now whose experiment gives you the basis of thermal stability of genetic material? Thermal stability का मतलब समझते हैं कि जो आपका genetic material है that will be stable at temperature. मतलब टेम्परेचर बढ़ने पे घटने पे आपका जो DNA है ना उसमें कोई चेंज नहीं आ रहा है. तो कौन था ऐसा whose experiment proves that genetic material shows the thermal stability? Tell me. Any idea? See, Hershey and Chase तो perform a blender experiment. इन्होंने कौन सा experiment किया था? Blender experiment. What he did in his blender experiment, इन्होंने just centrifuge किया था thing को, right? And he observed that, he proved that DNA is a genetic material with the help of blender experiment, right? Then Michelson and Stoll perform a density gradient experiment, right? They perform density gradient experiment. Okay, so they perform a density gradient experiment and he proved that he proved that DNA is a semi-conservative material. DNA replication is semi-conservative in nature. Then we have Jacob and Monod. So Jacob and Monod usually gave operon model. Okay, so he usually gave operon model, right? Lac operon model and all, which is related to the gene regulation in the bacteria. Now we have an option C, Frederick Griffith, who performed a ट्रांसफॉर्मेशन ऑफ जेनेटिक मटेरियल, ट्रांसफॉर्मेशन एक्सपेरिमेंट इन्होंने किया था. एंड व्हाट डज ग्रिफिथ विल परफॉर्म? Griffith chose, a bacteria named as Streptococcus pneumoniae. Okay, and this particular bacteria is responsible for causing pneumonia. So he chose two colonies of this, two strain of this bacteria, R strain and S strain. Right, now R is the rough strain, S is the smooth strain. R strain is non-virulent one. That means the R strain bacteria do not cause any disease, and the S strain is virulent, which causes disease. So when he inject R strain in the mouse, right, R strain bacteria in the mouse, nothing happens to the mouse and the mouse will alive. And when he inject the S strain in the mouse, then what happened? The mouse dies. Okay, again what he did, when he mixed both of them, R strain and the S strain, like when he mixed live R strain bacteria and he heat killed S strain bacteria. Okay, when he mixed heat killed S strain and R strain, heat killed means first he kill S strain bacteria in टेम्परेचर, उसको टेम्परेचर में रख के सारे बैक्टीरिया को मार डाला. किसको? एस स्ट्रेन वाले को. एंड देन लाइव आर स्ट्रेन के साथ उनको मिक्स किया एंड देन इंजेक्ट इन द माउस. सो अगर उनको इंजेक्ट किया माउस में तो अगेन व्हाट ही फाउंड दैट माउस डाइज, माउस मर जाता है. अदरवाइज अगर एस स्ट्रेन बैक्टीरिया को मार ही डाला है तब तो माउस नहीं मरता ना, माउस को तो नहीं मरना चाहिए था. इट शुड बी अलाइव, बिकॉज सेपरेटली जब उन्होंने हीट किल्ड एस स्ट्रेन को इंजेक्ट किया था तो माउस जिंदा था. तो लाइव आर स्ट्रेन से भी जिंदा है और हीट किल्ड एस स्ट्रेन से भी जिंदा है. सो व्हाट ही डिड, ही मिक्स्ड बोथ ऑफ देम एंड देन ही इंजेक्ट इनटू द माउस. तो इसके अनुसार तो उसको क्या होना था, इट शुड बी लाइव ना, बट द माउस डाइज. And when he took the blood sample, he found that now the R strains become virulent. Why? Because the S strain genetic material enters into the R strain. That means the transformation takes place. S strain genetic material enter into the R strain, and that R strain, live R strain, causes mouse death. So at temperature also the genetic material will be safe. So that is the stability, okay, proved by Griffith. Now the question is, short stretch of DNA used to identify complementary sequence in a sample. So what it is called? Probe, marker, primer, mini satellite. So here guys, the correct answer is your probe. What are probe? Probes are the short single stranded DNA और RNA sequence. ठीक है, ये probe है which is single stranded RNA, which may be a single stranded RNA or DNA which is complementary to the sequence and then used to hybridize with that original sequence. You can label a radioactivity also so that it can glow and you can identify. Okay, used to detect, probe is used to detect the complementary DNA sequence. What is the use of marker, primer and mini satellite? So markers used to identify organism or particular gene. Okay, marker used to identify particular gene, right? And the primer is what? Remember, the primer is during the replication of DNA, you need a primer. That primer will attached with the double stranded DNA. Primer जो है DNA के साथ attach हो रहा था and then the DNA polymerase enzyme will come and attaches to the primer and then started adding nucleotide. So primer is the short sequence of RNA. It is a short RNA sequence, right, used to start the DNA polymerization. Short RNA sequence used to start polymerization. Okay. Mini satellites are what? These are the repetitive DNA repeated sequence. Mini satellites are the repetitive DNA sequences. So DNA में कुछ sequences होते हैं जो कई बार repeat होते रहते हैं. Like ATG. Many times you have that repetitive sequence. So that repetitive sequence is called as a mini satellite. Now look at the question number 6. Select the incorrect statement. So now this question is related to your population genetics Hardy-Weinberg equation, right? What it is given, p square plus 2pq plus q square is equal to 1. This is the binomial expansion of p plus q whole square. Option B is saying when frequency measured differs from expected value, the difference indicate the extent of evolutionary change. Okay, let me explain you first of all the Hardy-Weinberg equilibrium. According to Hardy-Weinberg equilibrium, now you know what happened. He suggests that in a population, right? In a population, the genotype and the allelic frequency will be constant, and the same frequency of allele and the genotype will pass from generation to generation. तो एक generation से दूसरे generation में ये जो allelic frequency है and this genotype frequency is passes from generation to generation. Okay, so you can say that कि ये मतलब ऐसा ना कि constant है, like suppose in a population everyone is having black hair. Okay, so the allelic frequency of having black hair and the genotype of frequency of having black hair is constant in that population. Now the same frequency transfer to the next generation. तो जो next generation आएगी उनके भी सारे black hair होंगे. तो ये Hardy-Weinberg का equilibrium था. But the Hardy-Weinberg equilibrium got disturbed due to mutation, due to natural selection, due to genetic drift, right? So these are the causes, these are the factors which which changes the allelic and the genotypic frequency. Okay, due to which what happened? The equilibrium got disturbed. So now you have to select the incorrect. So P plus Q is correct. ये पहला statement तो correct है. Second statement is when frequency measured differ from expected value, ठीक है? So whenever you always this frequency is equal to 1, allelic and genotypic frequency is equal to 1. So the frequency what is measured expected value से 1 से अगर हमारी frequency change हो रही है, right? So that indicate the extent of evolutionary change. So that means कि if you have any change मतलब कुछ तो evolutionary change हुआ है. ये भी correct है. Hardy-Weinberg principle says that phenotype frequency in a population are stable and are constant from generation to generation. So this is wrong, right? You have to select the incorrect. So this is incorrect because Hardy-Weinberg principle says phenotypic, not genotype or allelic frequency, right? Genotype or allelic frequency should be constant. Okay. The gene pool, which is the total gene and the allele, remain constant. So ये भी correct है. So here the incorrect statement is your third one. Okay. Then the next is albinism is known to be due to autosomal recessive mutation. ऑटोजोमल रेसेसिव म्यूटेशन का मतलब क्या हुआ कि एल्बिनिज्म एक बीमारी है जो ऑटोजोम, वी हैव टू टाइप्स ऑफ क्रोमोसोम, सेक्स क्रोमोसोम एंड ऑटोजोमल क्रोमोसोम्स, राइट? तो ऑटोजोम वाले क्रोमोसोम पे रेसेसिव एलील, सी एवरी जीन के लिए, हर जीन के लिए हमारे पास 2 एलील होते हैं, 1 डोमिनेंट एंड 1 रेसेसिव एलील. तो जो एल्बिनिज्म है ये ऑटोसोम के क्रोमोसोम पे रेसेसिव एलील में म्यूटेशन के कारण होता है, ठीक है। द फर्स्ट चाइल्ड ऑफ द कपल with normal skin pigmentation was an albino. What is the probability of their second child will also be an albino? तो क्या probability होगा कि उसका second child भी albino हो? See here, it is caused due to the recessive allele A. So the couple is normal. So what happened? Here the couple is having normal. See, the first child of a couple with normal skin. That means कि हमारे जो couple हैं वो normal हैं, ठीक है, but has an albino child. लेकिन उनका child albino है. तो अगर suppose करो हमारे जो पेरेंट होंगे अगर कैपिटल ए होंगे तो वो नॉर्मल है, कैपिटल ए मदर भी होगी तो नॉर्मल है, बट नाउ दे आर हेटेरोजाइगस, तभी उनका पहला बेटा एल्बिनिज्म है। मतलब जो हमारे पेरेंट होंगे दे मस्ट बी हैविंग जीनोटाइप लाइक दिस AA and AA, so they are not homozygote, they are heterozygote. Okay, AA and AA. So when you cross, what you will get? AA, 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 AA. So this you will get. Okay. So now what happened? This is what? Capital A is normal. Here you have a normal child, right? Capital A, small a is the carrier one. ये कौन है? Carrier. And this AA, small AA is albino. Albino. So you can say this is 25%. Okay. These two 50%. And again this is 25%. So now what happened? You have 25% normal child, you have 50% carrier child, and you have 25% albino child, right? So question क्या है? What is the probability of their second child will also be albino? So the answer is your B, 25%. Okay, understood? So it's not like if you are going with MCQs, so very easily you can do. See, all the MCQs are concept based. It's not like you have to revise, you have to just memorize the fact. फैक्चुअल तो कुछ नहीं है, ये सब मेमोरी पे बेस्ड है, कॉन्सेप्ट पे बेस्ड है, तो यू हैव टू अंडरस्टैंड ईच एंड एवरी टॉपिक वेरी नाइसली, राइट? नाउ नेक्स्ट क्वेश्चन, मूव टुवर्ड्स द नेक्स्ट क्वेश्चन. In cricket species, the sound produced by rubbing the wings or legs together play a crucial role in attracting mates, right? So cricket species, you know, it is a kind of insect. Cricket जो टी 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 आवाज निकालते हैं ना, that will be the cricket. So the cricket species, right, what they did when they just rub their legs and when they just rub their wings na, they produce sound. So while they're rubbing their wings and legs, they produce the sound, and that sounds attract their mate. मतलब अपने पार्टनर को अट्रैक्ट करने के लिए they are just producing the sound. Any change in the morphology of the leg could potentially affect their ability to produce sound. अब अगर उनके लेग में कोई म्यूटेशन हो जाता है तो ऑब्वियसली कोई चेंज हो जाता है, तो साउंड जो प्रोड्यूस हो रहा था, दैट साउंड विल बी द प्रोडक्शन ऑफ साउंड विल बी चेंज. सो अ म्यूटेंट क्रिकेट हैज अ थिकर हाइंड लेग. तो अभी जो म्यूटेंट क्रिकेट है ना, उसका जो लेग था पीछे वाला वो काफी मोटा हो गया है, हाइंड. So what do you expect for this cricket species? So now read the option and think and tell me the answer. See, what would you expect? The leg mutation will not lead to speciation if they diversify into new habitat. Okay, let me explain you the concept, then we'll read the options. See what happened initially when you have no mutation, when the cricket has no mutation, then what happened? Normally the sound produced and the mates attract. But now what happened when the leg gets mutant or mutated? So now the leg becomes thicker. So initially जैसा आवाज प्रोड्यूस हो रहा था क्रिकेट से अभी नहीं प्रोड्यूस होगा. So now they are not able to attract their mate. Okay, so अगर suppose करो you have a population, in a population of cricket few crickets becomes mutant, right? Others are non-mutant, so others can create the sound, attract the mate, reproduce, fertilize and reproduce their progeny. So उनका तो population चल रहा है, generation आगे बढ़ रहा है. But what happened to those species, those who are not able to create that particular sound? Then what happened? They, they are not able to attract their mate, they are not able to fertilize, they are not able to reproduce. तो you can say कि अभी उसी population में there are few species which are not able to reproduce. So that is what you can say कि एक population में reproductive isolation हुआ है. In a population you can see a reproductive isolation, and slowly what happen, they will be created into a new species. मतलब अभी क्या हो जाएगा, वो speciation हो जाएगा, वो बाकी सबसे अलग हो जाएंगे. Now they become another species which are not able to reproduce, which have a thicker hind legs and which are not able to produce that sound. So this causes speciation due to reproductive isolation. Speciation मतलब क्या होता है कि नया स्पीशीज बन जाना, ठीक है, म्यूटेशन की वजह से. सो नाउ स्पेशिएशन किसकी वजह से हुआ? ड्यू टू रिप्रोडक्टिव आइसोलेशन. ओके, सो हियर द ऑप्शन इज the leg mutation will not lead to speciation. Wrong. The leg mutation will have little effect on the external feature. Wrong. Leg mutation will have no effect on behavior. Wrong. The leg mutation might lead to reproductive isolation and speciation due to the effect. So this is the behavioral reproductive isolation. Okay. So next question is your from health and diseases. Okay, here you have a plasmodium in a pathogen. Okay, so plasmodium is what? It is a pathogen that causes malaria. So we all know that plasmodium species causes malaria, and you have to identify the correct sequence. So here the 4 stages are given of malaria. Stage 1 is the stage of pathogen as it transferred by vector bite. तो पहला स्टेज क्या है? पैथोजन जब बाइट करता है हमारा वेक्टर, मॉस्किटो इज द वेक्टर. सो द फर्स्ट स्टेज ऑफ पैथोजन एज इट ट्रांसफर्ड. तो पहला स्टेज है ट्रांसफरिंग ऑफ पैथोजन वाया बाइट ऑफ वेक्टर. Second stage is first site in the host body where the pathogen infect and proliferate. तो जैसे ही पैथोजन एंटर किया, देन वो अपने पहले वो किस जगह पे जाएगा, किस जगह को इन्फेक्ट करेगा हमारी बॉडी में, एंड इट स्टार्ट पॉलिफरेटिंग, इंक्रीजिंग इट्स नंबर. देन सेकेंड साइट, अभी वहां से वो बाहर निकलेगा, रीचेस टू द सेकेंड साइट ऑफ द होस्ट बॉडी वेयर पैथोजन इन्फेक्ट and manifest clinical symptoms. So in the second site they shows the symptoms of malaria, and the fourth stage of pathogen as it transferred to a new vector. So you have to select the correct statement sequences. Understood the question? See, सो द फर्स्ट स्टेज ऑफ पैथोजन एज ए ट्रांसफर्ड, गैमेटोसाइट तो होंगे नहीं, तो ये और ये ऑप्शन तो खत्म. नाउ इट शुड बी ए और डी. ए और डी में से कोई भी हो सकता है. स्पोरोजोइट्स आर द फॉर्म ऑफ पैथोजन विच विल एंटर वाया मॉस्किटो बाइट. तो जब मॉस्किटो बाइट करती है तो प्लाज्मोडियम जो है स्पोरोजोइट्स के फॉर्म में हमारे ब्लड में एंटर करते हैं, राइट. देन दिस फर्स्ट स्टेज अभी हमारे ब्लड में एंटर करके वो कहाँ पहुंचते हैं सीधे? दे रीच्ड टू द लिवर. वो हमारे लिवर के हिपैटोसाइट सेल्स में पहुंचते हैं. सो स्पोरोजोइट्स, इरेथ्रोसाइट्स, नो, लिवर इन्फेक्शन. सो यहीं पे हमें आंसर करेक्ट मिल गया. दे रीचेस टू लिवर. Then inside the liver they converted into the merozoites, increase their number, and then what happened? They enters into the blood vessels and then infect the red blood cells which is called erythrocyte, and then there you will get the symptoms of malaria, and after that again you will get the gametocyte. This gametocytes will again suck by the mosquito. ठीक है, and ये सारा sequence होता है. So fourth one the correct one. Now the next is, which mRNA will be translated to a polypeptide chain containing 8 amino acid. ठीक है, आपको बताना है which mRNA will be translated to a polypeptide chain. इनमें से कौन सा mRNA जो है सीक्वेंस पॉलीपेप्टाइड की चेन में बदलेगा जिसके पास एटलीस्ट 8 अमाइनो एसिड्स होंगे. See, a polypeptide having 8 amino acids means it has 8 codons before the stop codon. Okay. Eight codon before stop codon, right? So what are the stop codons we have? UAA, UAG and UGA. What are the stop codons we have? UAA, UAG and UAA, UAG and UGA. UA, UAG and UGA. Okay, these are the stop codon. And which one is the start codon? AUG is the start codon. See, so all started from AUG. This is the start codon. Right. Now आपको ये देखना है कि इन stop codon के पहले 8 codon होने चाहिए. ठीक है. So now let us see the first one. UUA, one codon. AUA, another codon. Then here, see, ये तो यहीं पे रुक गया. You know, see here, you have UAA or you can say you have UAG. So now you have a stop codon initially only. तो ये तो नहीं हो सकता. Then now moving towards the second one. ठीक है. AUG, AGA, CGG, ACU, GCA, UUC, CCA, ACC. So now see UGA. UGA is the stop codon. So before UGA, how many codons you have? 1, 2, 3, 4, 5, 6, 7. And AUG is also a codon. ठीक है, ये भी तो एक codon ही है ना. तो you can say that कि ये हमारा correct answer हो गया. ठीक है. Here also see you have UAG initially, पहले ही आ जा रहा है. 7 से पहले, 8 से पहले ही आ जा रहा है. ठीक है. So like this, here you have UAA before 8. So like this you have to check कि stop codon के पहले पहले कितना आता है. In order to isolate genetic material of a bacterium, the cell must be treated with. तो जेनेटिक मटेरियल लाइक आइसोलेट अगर करना है आपको जेनेटिक मटेरियल किसी बैक्टीरिया से, तो सेल को सबसे पहले हम किससे ट्रीट करते हैं? तो यू नो द सेल वॉल ऑफ बैक्टीरिया इज मेड अप ऑफ पेप्टिडोग्लाइकेन, राइट, एंड दैट कैन बी digested with the help of lysozyme, right? Then ribonuclease, protease, chilled ethanol. So these are the sequence. Cellulase, if you have a plant cell, if you want to isolate DNA from plant cell, then you have to treat with the cellulase first. If you have a fungal cell, if you want to isolate DNA from the fungal cell, you have to treat with the chitinase, right? So this is for plant, this is for fungi. Okay. Integrate pest management involve. So integrated pest management, the use of pesticides. Integrated means like you are doing some modification, you have done some modification in the pest. So using pesticides, insecticides judiciously, using biocontrol agent, engaging in organic farming. So here integrated pest means both 1 and 2. Like with the judge also you will get the permission, and you are using the biocontrol agent. Now you have a assertion and reason type of question. So till now you have done with the MCQ correct answer based question. Now you have assertion reason. Assertion reason में ना you have one assertion A and the reason R. तो answer हमें कैसे करना होता है? Both as you have to read the statement, both the statement, one is assertion, another one is the reason. So you have to read them. Then you have to select that whether the assertion and the reason statement both are correct, and reason is the correct explanation of assertion. So both A and R statement true, R is correct explanation of A. Second is both A and R is true, but R is not a explanation of A. Statement दोनों correct है, but R is not explanation of A. Then A is true, assertion is true, but reason is completely false. So this statement is completely false. A is false, assertion is false, reason is true. So like this you have to answer. ठीक है? So now in your board exam, again how you have to answer, how you have to write down in the copy. So simply 13, whatever will be the option A, B, C, D, these are the options. So suppose for first question, option B is the correct one. For example, for first question, option B is correct. So how you have to write down? 13 answer B and both A and R true like this. So उसी तरह आपको पूरा लिखना है. ठीक है? Now let us go with the question. Assertion says the ability of the pistil to recognize the pollen is the result of continuous dialogue between pollen and pistil. Electrical reason says that electrical dialogue मतलब interaction. Electrical dialogue allows only compatible pollen to germinate. Now see, here the statement first is completely correct. That means you can say that the assertion is correct. The ability of the pistil to recognize the pollen is the result of continuous dialogue correct है. तो pollen और pistil में interaction होता है, right? So pistil will recognize whether the pollen is of same species or not, and when the pollen get that signal, then only it attaches the pollen towards the stigma. Okay, so there will be a chemical signal releases. Okay, due to some biochemical signal, the pistils are able to recognize their pollen, known as pollen pistil interaction. Electrical signal is not, यहाँ पे electrical signal नहीं होता है. Okay, so you can say that option for first question, right? Option C is A is true, but reason is false. So here C is the correct one. Okay. Second says some organisms are better adapted to survive in an otherwise hostile environment. ठीक है, adaptive ability is inherited and as a genetic basis, so this is correct only. Okay, जो organisms हैं, few organisms can adapt to their situation, to their environment because they have some inherited capability. उनके पास कुछ inherited ability होती है due to which they can adapt. So here both are correct and reason is the correct explanation of the assertion. Then we have 15. Excess dose of coke or crack produces a sense of euphoria, increased energy and causes hallucination. It interferes with the transport of dopamine. So here again, excess dose of coke produces a sense of hallucination. करेक्ट है. इंटरफेयर हमारे डोपामीन को इंटरफेयर करता है, ये भी करेक्ट है. ओके, देन assertion says Rosie was the first transgenic cow to make more nutritionally balanced milk for consumption by human baby. The milk of Rosie contained human beta-lactoglobulin which made the milk rich in protein. So see, ये जो Rosie cow है, this is a transgenic cow which is made to produce a nutritionally high quality of milk which is similar to the mother milk. Okay, so the first statement is correct. First transgenic cow है, nutritional balanced milk बनाती है. Reason कह रहा है Rosie cow contain human beta lactoglobulin which made the milk rich in protein. No, this is not correct. So here you can go with assertion is true, reason is false. So C is the correct one. Okay. So like this, you have solved 16 questions, right? 16 marks questions. Now, let us go with more questions. See, this is your section B and here you have 2 marks question. तो अभी 2 नंबर का जो क्वेश्चंस होंगे ना, मैक्सिमम यू हैव टू राइट डाउन 4 to 5 लाइन्स. ठीक है, डिपेंडिंग अपॉन द क्वेश्चन. समटाइम यू विल गेट 2 क्वेश्चंस स्टेटमेंट वाइज, सो सिंपली यू हैव टू राइट डाउन द आंसर. ओके, जैसे यहाँ पे देखो क्वेश्चन है, ड्यूरिंग आर्टिफिशियल हाइब्रिडाइजेशन इट इज इंपॉर्टेंट टू एंश्योर दैट ओनली डिजायर्ड पॉलन ग्रेन आर यूज्ड फॉर पॉलिनेशन एंड हाउ इट इज एंश्योर्ड, राइट. तो अभी यहाँ पे कह रहे हैं आर्टिफिशियल हाइब्रिडाइजेशन में, ठीक है, it is important to ensure that only desired pollen grains are used for pollination. You know artificial hybridization, right, where you want कि आपको पता है कि कौन से pollen को किस पे hybridize करना है, ठीक है. So here you need to do 2 things, emasculation and bagging. So in this question you have to explain 2 things. What are the 2 things? The emasculation and the bagging technique. यही आपको explain करना है इसमें. तो अगर ये term आप explain करेंगे then only you will get the answer, because while emasculation what happen? See, in case of bisexual flower you have both pollen, you have both anther and the pistil in a same flower, so there will be a self fertilization, right? You cannot stop them, so you have to cut the anther, right? That is called emasculation. The removal of anther from bisexual flower is the emasculation. Then you have to cover the flower, known as bagging. ठीक है, आपने उस फ्लावर को कवर किया, उसको क्या कहा गया? बैगिंग कहा गया. So by the use of emasculation and bagging, right, it is important to ensure that only desired pollen. So now whatever the pollen you want to hybridize, you can do it. So this is with the help of emasculation and bagging you can do it. Now why is manual transfer? Now here this question has 2 options. Either you can go for this or you can go for this. So why is manual transfer of desired pollen grain relatively easy in pea plant for hybridization, but in wheat pollen grains are often utilized from pollen bank? So what is the reason behind that? Any idea? See, pea is the bisexual flower. So in pea plant, the manual emasculation is very easily, right? And the pollen is also viable for month. Pollen grain भी 1 महीने तक viable रहता है. So that is easier. But in case of wheat, what happened? The pollen grains loses its viability within a half an hour. That's why it is required to take it from the seed bank. How is the rate of initiation of RNA polymerase at a given promoter in a transcriptional unit of prokaryotes regulated? See, यहाँ पे 2 चीज पूछा है, ठीक है? How is the rate of initiation of RNA polymerase at a promoter in a transcriptional unit of prokaryotes regulated? तो बेसिकली हमारी जो ट्रांसक्रिप्शन यूनिट होती है, इट हैज अ प्रोमोटर, स्ट्रक्चरल जीन एंड ट्रांसक्रिप्शनल दैट टर्मिनेशन पॉइंट, ठीक है? तो आरएनए पॉलीमरेज विल रिकॉग्नाइज द प्रोमोटर, but who will help them to recognize the promoter? Definitely there are some regulatory proteins which help the RNA polymerase to recognize the your promoters, so that they can initiate the transcription. Now these regulatory proteins are of 2 types, activator also and inhibitor also. So based on that regulatory protein, the transcriptional site started. अगर suppose करो inhibitory regulatory protein आ जाएगा, then what happen? The transcription will not start. Activator आएगा, then it will start the process. Okay, so this is how they regulate. So you have to answer like this. Try this question. The table below shows a hypothetical blood report of a patient. तो आपने भी blood test कराया होगा. ये blood report आया है where the total leukocytes, neutrophil, lymphocyte, basophil, eosinophil and monocytes are given. These are the value observed from your blood and these are the reference. इतना होना चाहिए and these are the values you will get. Looking at the value suggests which defense mechanism or immunity is affected and state how this defense mechanism provide immunity. Name the barrier with the least count and enumerate its role in providing immunity. See, here in this case, you are not taking any extra vaccines and all. So this is the innate immunity which is already present in your body. So which type of immunity here you have? Innate immunity. Okay, so innate immunity is a non-specific type immunity which is present from the birth, right? And here which type of barrier is? So see, here the monocytes should be 1 to 8, but here the monocytes becomes 0. So cellular barrier you have where the monocytes, with least count and it can phagocyte the destroy microbes. So here the barrier is cellular barrier. Okay. Now the next 20th question. Here either go for A and B. So till here we'll end the session now. In the part B, okay, we'll continue from the remaining portion. Okay, so I hope you have understood all the topics, you have revised all the topic. Abhi sara yaad aa raha hoga. Now in the next class or you can say in the next session, we'll continue the remaining revision questions. All right, so let's end the session here only. Meet you in the next class very soon.
अब एग्जाम की तैयारी को और बेहतर करने के लिए हमारे चैनल को लाइक, शेयर और सब्सक्राइब करना ना भूलें।